Hello everyone, today I would like to introduce you a DNA binding site prediction method, DeepDISE. We all know that most of the proteins perform their functions through binding and interacting with other molecules such as small compounds, nucleotides, proteins, lipids, and so on. Therefore, to identify those binding sites is a very significant task. There were two reasons why we would like to create a new DNA binding site prediction method. Number one, DNA binding proteins play central roles in many cellular processes, such as DNA replication, repair, transcription, regulation of gene expression, DNA stabilization, and degradation. Number two, we have reliable small molecular binding site prediction methods. For example, we created a method called LISE using ligand interacting and the binding site enriched protein triangles. The results are good. You can see that those empty grids colored in from blue to red, predicting the potential ligand binding site. And this and this. In addition, deep learning technology has been mature and widely used in many different prediction methods. So we would like to integrate deep learning technology into our previous binding site prediction method. To avoid overfitting, we prepared 3 independent data sets. We collected 274 PDBs of DNA binding proteins for training sets and downloaded 2 test sets published in previous studies containing 62 and 224 PDBs respectively. In the 274 PDB training sets, about 90% of the PDBs were used to train the deep learning model, and the rest of the 10% of PDB were used to validate the prediction performance. The rest 2 test sets were used to make sure there's no overfitting and to compare the outcomes with other existing methods. We wrote a program to assign atom types on the atoms of target proteins. If an empty grid was within 4 angstrom from any binding DNA atoms, it would be assigned as a binding site. Otherwise, it would be a non-binding site. The input data simply contains assigned atom type and the X, Y, and Z coordinates of each protein atom. DeepDISE is a fully convolutional neural network. Every DNA binding complex was rotated into 24 orientations to train our CNN. Just like the picture here, the output data is continuous. We have to set a threshold to determine which grids are the predicted binding sites, or clustered the high-scored grids into a patch like this. We can check this figure, although there are still some false positives here and false negatives here. We can say that the predicted sites are pretty much accurate and precise. And we can check these 3 tables for 3 data sets. The results of the training sets and the validation sets are similar, as well as the outcome of 2 independent test sets published by previous studies. This shows the robustness of DeepDISE. Compared to the outcome of previous methods, the DeepDISE performance is comparable or even better. In this study, we created a deep learning-based method to predict the DNA binding site on the target proteins. We demonstrated that a deep learning method only using 3D protein structure plus atom type of protein atoms can well predict DNA binding site. And this approach should be applied to other predictive models in the future. Thank you for listening. My name is Zhong-Ru Xie. If you are interested in our research, please feel free to contact us. Thank you.